Right. Hello, everybody. Welcome to Human Genetics SIL3008. In this series of videos, I'll be talking to you about Chapter 7, which is analyzing the structure and expression of genomes from the book Human Molecular Genetics, Tom Strachan and Andrew P. Reed. And this is the latest edition, which is 2019. Right, so in chapter 7, it starts off telling you about the importance of, or how, or rather, how the genome was analyzed. And it says, in the 1980s and early 90s, when they started off with the Human Genome Project, they thought that, they means the scientists, they thought that was the biggest challenge of a lifetime. But as it turned out, now getting the sequence is the beginning. Is that was the easy part. Understanding how all these sequences and how the genes work, that is the bigger challenge at the moment. So first things first, genome structure and genome projects. When you talk about genome analysis, if you look at the text, it says that if you're analyzing bacterial genome, it's not such a big a problem, because when you do DNA extraction, you do get just that piece of DNA. Unfortunately, for metazoans or multicellular organisms, I mean, in humans, let's just take humans, we have 23 pairs of chromosomes. So whenever you, a simple question that I may want to ask you is that if you isolate DNA and you are particularly interested just in a particular region in chromosome 7, so how do you actually get chromosome 7 out, and be able to analyze from that lump of DNA? So that was one of the biggest challenge. And so one of the things that most of the strategy towards being able to sequence the genome is, first of all, you need a framework. And by framework, you need to be able to sort out all the different chromosomes. And in order to share the load at that time between different laboratories in the world, they must be able to have to create libraries where individual chromosomes can be obtained and can be analyzed individually. So there are a few technologies mentioned later on, as you can see, but once you have that particular piece of fragment in this section, if you look at these notes in this section, it talks about the shotgun sequencing. What that means, if you have to know how a shotgun shoots. If you use a normal rifle, you shoot with a bullet and then you get one mark. But if you use a shotgun, it sort of gives you a bigger coverage. It just gives you sort of a wider effect. So this is when they say shotgun sequencing, it means that you don't sequence one point at a time. This is the DNA. You don't sequence from that point and then move to the next and then move to the next and then move to the next. You don't do that. But shotgun sequencing is that you shotgun everything, you sequence everything in one go. Well, of course, what you need to do then, if you look at the figures, is that you'll need to chop this up first, right? Sequence it and then piece them back together again. Of course, it won't be much of a problem if you're talking about bacterial DNA, because you only have one chromosome. But if you're talking about human DNA, then you have 23 pieces, 23 pairs of chromosomes. If you chop them up, it's like, when you have, I mentioned this in class, where you talk, the analogy would be playing with jigsaw puzzle, right? In which you actually have 26 different puzzles all mixed together, and you need to be able to sort individual puzzles out again before you can actually start to sequence it. All right, so that was the challenge. How do you uh, separate all these individual chromosomes so that you get a framework, a library, a resource that is not contaminated by other chromosomes, right? The strategy for cloning is given in figure 7.1, generating clones with overlapping DNA insert. This tells you a piece of DNA fragment is cut with an enzyme, but if you cut with the same enzyme, you'll get the same fragment. The idea is written in rather small sections. Here it says, if you can follow my mouse, it says partial digestion. Partial digestion means it doesn't cut all the sites. So assuming, if you understand this figure, number 1, number 2, number 3, number 4 is 4 copies of the same chromosome. I mean, if you have enzymes that is given in the full amount, it will cut at all the same places in all fragments. But because you are given a limited amount of enzymes, so each fragment will be cut differently. You don't have enough enzymes. So and the result is that you will have these different fragments A, B, C, D, and so on. And these different fragments can be cloned into a suitable vector, right? And then what you have is a genomic DNA library. Okay. Now the next picture, figure 7.2, talks about the difference between a whole genome shotgun and a hierarchical shotgun. Well, the strategy is the same. That means you chop them up into smaller pieces and then you sequence. But because when you're dealing with human DNA, you have multiple different chromosomes. You cannot use the whole genome shotgun. Whole genome shotgun is doable for bacterial chromosome. When you isolate the bacterial chromosome, you have just one chromosome, and then you just chop it up, and then you just sequence everything, and then paste them back together again, no problem. But for human genome, or for any genome that is made up of multiple chromosomes for that matter, what you need first is to isolate the chromosomes. That part, this is where you see contig of large inserts there. I'll explain to you what contig is. Contig of large inserts, and then from there you do your whole genome shotgun. All right. So this is why it's called hierarchical. That means you need to do an extra step so that you deal with the multiplicity of different chromosomes. Okay. I'll try to keep this videos short, so on to the next video.